Much of the work I will describe here today has been done in the context of a new centre in the Department of Chemistry, of which we're part together with my colleagues, Professor Christopherson and Professor Michele Majuscola. And the idea is really to try and develop an interdisciplinary centre to look at some of the physical and chemical underpinnings of neurodegeneration. So that's sort of the general context for the work. And I'm actually not sure I've given a seminar in a mathematics department before, so this will be sort of a selection of our research, and a very biased selection where the focus will really be on sort of the detailed quantitative characterization. So that's the idea. But maybe first a couple of words of introduction. So the general idea is we're interested in proteins. And so why we're interested in proteins? Well, from a sort of physical scientist point of view, anything that does anything in biology is typically a protein. And the way that proteins are able to fulfill their biological roles is typically not on their own, but rather by binding to other proteins in order to form functional complexes. And if the proteins manage to find the correct partner, well, this is great, this gives the molecular machinery of life functional complexes. But of course, what can happen is the proteins can bind an incorrect partner, or they can even bind a correct partner but in an incorrect manner. And this then leads to pathological structures which are involved in a range of diseases, including the neurodegenerative diseases, to which I'll come back slightly later. But again, from a very basic point of view, this is sort of really the molecular basis for biological functionality, but not necessarily very easy to study using conventional methodologies. And this is partly because these complexes can be very diverse. Some of them are just formed very transiently, others are very tightly bound, and they're typically, in many cases, dynamic and also heterogeneous. So much of what we do is really trying to come up with new tools in order to really address these challenges and try and understand in a quantitative way how proteins come together in order to generate both function and malfunction in nature.
So on the picture on the previous slide, what is the length scale on the picture on the right?
So the length scales are typically nanometers. So the length scale here is also sort of order magnitude nanometers. So this is sort of one of these pathological amyloid structures I will come back to at a slightly later stage. So these are really sort of supramolecular length scales, which are intermediate between sort of atomic length scales, which are of interest in much of sort of, say, synthetic chemistry, and sort of the macroscopic world, which we sort of think of in terms of biology in the conventional sense. So this is really biology on a molecular level. And a particularly interesting and a practically relevant example of this self-assembly phenomenon is if proteins, instead of folding into their native state, so here's, for example, a representative picture of a native state of a protein, so this transitions from an unfolded chain into a folded native state happens when, within this polypeptide chain, there's non-covalent contacts which are formed between different residues along this polymer, and this directs folding into these types of native states. But of course, these same contacts here can be satisfied not only in intramolecular manner, but also in an intermolecular manner. And this leads to the formation of arguably sort of the simplest example of a supramolecular protein structure, which is for historical reasons known as this amyloid state of proteins. And it turns out that this type of state is actually accessible for a very wide range of different proteins. This was first pointed out by my colleague Christopherson in the Department of Chemistry many years ago now. And this really relies on the fact that the interactions here, which are shown in yellow, these are really backbone hydrogen bonds from a sort of chemical point of view, and they rely just on the common polypeptide backbone, which is shared between all proteins. So despite
that's a beta sheet.
That's a beta sheet. That's a beta sheet structure. Exactly. So there can be parallel or anti-parallel. There's quite a wide variety of different possible packings. But in principle, these are structures where the stability is really driven by these backbone hydrogen bonds. If you think about it, again, from a very basic point of view, there's sort of two main secondary structure elements that proteins adopt, the alpha helix and the beta sheets, which both satisfy backbone hydrogen bonding constraints. It's only the beta sheet which allows these constraints to be satisfied in an intermolecular rather than intramolecular manner. And again, that's at a very basic level why these amyloid structures can be formed from so many different sequences. And that's sort of what I have here, so that independently of the primary structure, independently of the native structure of the proteins, these species have very similar morphologies on sort of ultrastructural scales, and even down to the molecular scale, they are, as you pointed out, beta sheet rich. So despite being one of the simplest examples of supramolecular protein self-assembly, it's also one of the more sort of interesting in terms of practical implications for functional biology, but of course also aberrant biology. So a couple of examples here: functional systems which exploit this amyloid fold. So for example, many functional materials in nature have these types of amyloid structures as structural components. For example, many biofilms that bacteria secrete in order to sort of facilitate colony formation contain amyloid fibers as a structural component. Even the colon E. coli biofilms, and also not just in microorganisms, but even in mammalian systems. In fact, even in humans, there's examples of amyloid materials having functional roles. For example, this protein P17 forms catalytic scaffolds, which are active in the context of biosynthesis of melanin in humans. But of course, the major reason why there has been a lot of interest in understanding the molecular mechanisms of amyloid formation is because they're linked to human disease. And at some level, this is not entirely surprising. So if proteins, instead of being part of a particular type of biochemical network and being isosoluble, if instead of doing this, they self-assemble in an uncontrolled way in order to form these types of nanostructures, you can imagine this is probably not going to be very helpful for the functionality of the cell. But what's remarkable is that there's really a very diverse set of disease states, ranging from neurodegenerative conditions, Alzheimer's disease, prion conditions, Parkinson's disease, but also sort of 30 or so slightly more exotic diseases which are known as systemic amyloidosis, which affect very different parts of the body, so the heart, the liver, the spleen, the of course central nervous system here for the neurodegenerative disorders. And they appear on a macroscopic level to be entirely unrelated, but on the molecular level they are all characterized by the uncontrolled transition of proteins from the soluble states into these types of nanostructures. And this is sort of at a very basic level the process that we would like to understand. How does this happen? Why does it happen? And is there something that we can do in order to control this in a rational way, in order to prevent these increasingly prevalent and currently incurable diseases from becoming an even bigger burden on society than they are at the moment? So that's sort of the general introduction, and the sort of, yeah, please.
Naive question. How does amyloid differ from, say, actin? Actin is also a...
Right. So I think I will actually show some data to answer that question. But I guess at a very basic structural level, actin is not a beta sheet rich polymer. So on a sort of an ultrastructural level, they have very similar morphology. On a molecular level, they have very different structural characteristics. But it turns out that there's also some very basic physical differences, which I will actually, I think I will highlight one particular difference that we've been very interested in. So if I haven't answered your question in the next 45 minutes or so, then you should ask it again. And so the things that I wanted to sort of share with you here today, as discussed, are very biased selection of the types of activities that we're undertaking. I'm not going to be talking very much about sort of the biology and the medical aspects, but rather really focusing on the sort of physical side. And in particular, I'm going to be talking about kinetics, so the dynamics of this system. And even though this is a seminar in a mathematics department, I will show a couple of examples of new types of measurement strategies that we've been developing, where we are probing protein aggregation in very, very small volumes. All right, so for the first half, we'll talk a little bit about mechanisms. And so the way we got into looking at the dynamics of the system was really by asking the question, well, what exactly is the mechanism of this type of process by which proteins transfer from the normal soluble states into these fibrillar states. And so the way that this type of question is answered conventionally in chemistry is by looking at the kinetics of chemical reactions. And indeed, there's sort of what you could call a conventional recipe on how to relate macroscopic measurements to microscopic mechanisms. And the recipe essentially says you start off with a differential rate law. So this is sort of almost a bookkeeping exercise. So for example, for simple dimerization of, say, two A's coming together to form a B, we say, okay, well, let's keep track of the species A, and their concentration as a function of time changes with a rate which depends on the second power of A, the square of the concentration of A, because in this case it's a dimerization reaction. Now, of course, the conventional recipe says, well, this is not so convenient if I'm in the lab and I'm doing measurements. This is not really what I want to measure. I don't want to measure derivatives. What I measure in the lab is concentrations, and therefore integrate, and I get these types of integrated rate laws, and from these integrated rate laws I can evaluate global quantities, such as, for example, half-time. And it turns out that for the second order reaction, you get this type of simple, well-known behavior where the half-times, the time that it takes for half of my reactant to convert to the product, depends on the inverse power of, depends on the inverse of the initial concentration. So this is sort of a power of -1. So somehow, this power of -1, somehow that contains the information that there were two things that came together. And so chemical kinetics is really sort of the formalism that allows us to establish this connection. Okay, so this is the conventional recipe that's taught in sort of first year physical chemistry, that's been used for the past probably 100 years in many areas of small molecule chemistry. And so what we want to do is we want to take this approach and apply it to the slightly more complicated system of proteins coming together to form linear filaments. And so there's a range of things that we need to take into account. And so if we focus a little bit on the sort of series of microscopic steps which are required in order to have a meaningful description of this process, but we certainly need some type of nucleation phenomenon. So we need proteins to come together in order to form nuclei. This nuclei can grow by monomers adding on to the ends of the filaments. That's why they're filamentous structures, because the monomers add to the ends. If we worry about microscopic reversibility, as we probably should, we need to allow for monomers also to be able to detach from the ends of the fibers. And this was again something that my colleague Christopherson pointed out 10 years ago now, that is actually experimentally a key feature. Even though these fibers are actually very stable, this process does actually exist, and in many cases has important biological consequences. Because these structures grow from the ends, we have to keep track of processes which change a number of ends. So the simplest one of those is if a structure fragments. So here I have 2 ends, it fragments, I've doubled the number of ends, I have 4 ends, so I need to keep track of this. And of course there's other ways in which I can change a number of ends. So for example, if there's a nucleus that's formed on the side of an existing fiber, then this creates new ends. This is what we're calling secondary nucleation. All right, so now we can just sort of look at the dynamics of the system exactly in the same way as we would look at sort of simple chemical reactions. And so we can write down master equations, a partial differential equation for looking at the time evolution of the number concentration. And now, of course, instead of having just one species, we have an infinite number of species, so we just index them by j, and we keep track of what happens. And so this is now, as all master equations, this is essentially a bookkeeping exercise. So I need to go through these processes and figure out what goes where. And so, for example, if we start by, so we keep track of species of length j. So how can I form a species of length j? Well, if I have a species of length j minus 1 at time t, and I add one monomer to it, then it's going to be of length j. So I need to add it to my accounting here. And there's a factor of 2 here if we think that the filaments grow from both ends. And the rate constant here I'm calling k plus. The factor 2 is actually non-essential, so if the filaments only grow from one end, of course, I can subsume this 2 into the rate constant. But now, of course, I have to do the accounting properly. So I have to also make sure I remember that the structure, which was of length j, if I add a monomer to it, is no longer of length j, because it's of length j plus 1, so I have to remove it from the pool. And then the same thing for if I do the same accounting exercise for the offerings, I get something like this. And of course, what's even more interesting is the fragmentation processes, because they couple equations from 1j to the, so these only couple locally, so these couple from 1j to the next. The fragmentation process is also coupled globally, because any filament of a size larger than j can fragment at exactly 2 locations in order to yield a fragment of length j. So that we'll have to keep track of all the filaments which are larger than length j. These are the secondary nucleation process, and this is the primary nucleation. Okay, so far this is really just, you can always write down a master equation of this type if you have a molecular description, so we can write this down. It's not very useful for the time being, because this is an infinite set of coupled nonlinear differential equations, so it's not going to be very easy to compare with experimental data, which is what we want to do.
You also are allowing branching, so then you have 3 ends and 4 ends. And
yeah, that's right. So this is sort of, so this secondary process here is sort of an example of of branching. So in principle, these structures may or may not detach from the ends. One can actually write a very similar equation. So here I've actually written the fact that so here we're assuming that the filaments actually detach when they nucleate off the sides, but you can write a very similar equation where you essentially assume that the filaments stay attached to the parent fibers. So it doesn't really change qualitatively the formalism. And in fact, I think I might have one example slightly later for actin, where this is actually part of the functional role of actin. There's cofactors which favour branching.
You see the parameters N2 and NC, are they considered known a priori, or do you consider
Right.
them to be determined?
Right. So great question. So I haven't actually defined any of the parameters in any detail. So these are essentially reaction orders. So these are the NC is the reaction order for the primary nucleation. For certain simple cases, for example, if it's an elementary reaction, or if this process follows classical nucleation theory, this reaction order is actually related to the nucleus size. But for many other cases, it's in fact not related at all to the physical size of the nucleus, and it's simply a reaction order. And it's typically not known a priori, so this is also something that one will have to worry about. And of course, there's no reason why this reaction order would be the same as the one for the secondary process, and indeed, in general, it's not. So that highlights another issue. There's a lot of free parameters.
So there's also some equation for MT.
Right. Yeah, exactly. That's a great point. So that's I haven't shown that here, but you need that. And in principle, the way we obtain that is by enforcing conservation of mass. So in principle, we're saying the total number of monomers is constant. So in principle, what you do is you sum on both sides of the equation, and then you see whatever is consumed by the filaments larger than the monomer. This is removed from the monomer.
You don't consider that two filaments can also get together?
Yeah, also fantastic questions. I made a big point about stressing microscopic reversibility, and I left out the inverse of the fragmentation. So in principle, you can include that process in exactly the same way. I've left it out here, partly because otherwise the equation doesn't fit on the slide, but also on a more sort of serious level, because it actually turns out that for most of the systems that we're interested in the laboratory, that rate is very, very small, and it's essentially almost not observable. The reason for that is that in order for these filaments to anneal, the polypeptide chains are relatively long, and so the interface is actually quite complicated. So the filaments would have to come together with perfect alignment, perfect torsional alignment, and that's actually a very, very slow process. So it's essentially not observable in experiments. There's other filamentous systems where that's actually very important. So, for example, the things that have been looked at earlier in terms of colloidal aggregation using sort of the Smoluchowski framework, and in fact, indeed, living polymerizations are something that Mike Cates has looked at extensively in the 1980s, 1990s with particular amphiphile surfactant molecules. So there are cases where this process is important. It turns out for protein filaments, in particular amyloid fibers, the rates are very low, so it's not considered here. But in principle, there's no problem in including it in this framework. And okay, but so the challenge we have is we have to. So this is clearly fine, and this is essentially a bookkeeping exercise. But how do we actually extract some useful information out of this? And really, what we want to get some idea of the integrated dynamics, so the integrated rate law. And there's sort of a variety of ways in which one can approach this problem. And I'll just show a couple of examples. So the first thing that one can do is one can say, well, let's not worry about this full distribution. Let's only worry about things we can actually measure. And it turns out in the lab, what you can measure is you can measure moments of this distribution. So you typically measure number concentrations. That's a 0th moment of this distribution. Or you measure the mass concentration, which is the first moment of the distribution. And so now if you take the Maxwell equation of the previous slide and you do these sums, so you sum from 0 to infinity on both sides, and you do the usual switching of sums and derivatives, etc., things simplify quite a lot. And in fact, it turns out that to a very good approximation, the first two moments form on their own a closed system of equations. So that's useful.
Sorry, did you really mean to say sum from j equals 0 or from j equals 2?
So in principle, you actually sum from the critical nucleus size.
So there's some conservation law for if you really were to sum over all j, right?
Exactly. So in principle, we're enforcing conservation of mass, and so the sum of all of the molecules. We're not creating any molecules or destroying any molecules. In principle, one can generalize this framework to do exactly that, because in a living cell, there's protein synthesis, there's protein degradation, so we have actually worked on generalizing this framework. I won't talk about that here today. So this is really going to be conserved mass. And so the very good approximation, these first two moments decouple from the rest, and we get sort of equations of this type. And these have sort of actually sort of a fairly intuitive interpretation. So if we're asking how does the number of aggregates change, well, it changes if aggregates fragment, so that's why there's a fragmentation rate. So if I fragment aggregates, I'm changing the number of aggregates. And it turns out that the fragmentation rate is going to be roughly proportional to how many aggregates I already have in the system. What is the mass of aggregated material in the system, because that's related to the number of bonds, which can potentially fracture. That's why I get terms like this. And again, here, the majority of this is the change in the mass concentration of aggregated material, and the majority of the mass is consumed by monomers adding onto the end of the aggregates. If the majority of the mass were consumed by formation of new aggregates, we wouldn't get linear structures. Then we'll just get lots of small clumps. And so that's sort of what's captured by this kind of equation here, because this is the number of fibers and this is the elongation rate. So this sort of tells us that's how the monomers are consumed. But this really, you can just derive this formally from the master equation, just to make the point that these equations do have qualitative interpretations, yes?
And what kind of approximation do you do to decouple the equations from higher order?
Yeah, so essentially there's a couple of things we neglect. So these tend to be sort of terms which are which depend on sort of inverses of rates which we don't consider. So for example, the inverse of the nucleation process is another one which we sort of have, which are left out on the scheme, and that's something that you have to neglect in order for this to work, basically. And then, I mean, there is a weak coupling, for example, between this one and the second and the second moment. Okay, all right. So this is a good start. These are still, however, non-linear differential equations, because again, this m here is related to this m here because of conservation of mass. So this is non-linear, and they're coupled. And we've so looked at a variety of ways of dealing with this problem. A particularly simple and one which I'll start off with, and which has sort of a nice qualitative interpretation, is actually looking for self-consistent solutions. And this we found works actually very well and is easy to implement. So you can take this sort of set of moments here, first two moments, and essentially you can formally integrate on both sides, and you get something like this. Here nothing has happened, just formally integrated on both sides. And so I end up with a self-consistent form. So here I have sort of this two-dimensional vector, 0th moment, first moment, and I have some function which operates again on this, on these first two moments. Okay. All right. So how do we solve? So this isn't the solution. This is just the massage the equation into the self-consistent form. But of course, so in principle, this is sort of a summary. So I have some operator which operates on this space of the first two moments. This is operator here, and it gives me back these two moments. Great. So this is actually a fixed point equation, right? So these are easy to solve. So we just use the Banach approach. And so the way we can solve this is by just simply iterating this operator on initial guess. And of course, if we have all the usual, sort of conditions, it has to be a contraction, etc., then we're actually guaranteed to get the correct solution. So this is the way that we operate.
How did N2 drop out of your iteration scheme?
That's a very good question. So it's not going to drop out entirely, because it's actually going to reappear. It's going to reappear here. Or more specifically, the reason why it dropped out was because we had there's sort of an additional simplification, which you don't have to make, which I made here in order to keep the equation slightly more manageable. So essentially, we're really saying we've left out these corrections here, as well as these ones here in front of this dominant term here. So this is really saying that we form filaments, that the major sink in terms of the mass of monomer consumed is really in elongating structures rather than forming new nuclei. If it were the other way around, if the majority of the mass went into forming new nuclei rather than elongating existing ones, you wouldn't have filamentous structures, because all of your they would essentially be similar to diffusion limited aggregation. It would just have essentially lots of small structures. Okay, all right. And so now, in order for this to work, of course, we need to have an initial guess, which is somewhere as close as possible to what we need, such that we're sure that things will contract around that. So the way we do this is actually by linearizing the equation, looking for linear solutions. And this is essentially equivalent to neglecting conservation of mass. So the reason why these equations are so non-linear is really because we're keeping track of this fact that when monomers are used up, they're no longer available for any of the other processes. So if you forget about that, if essentially, back on this slide, if I say that the monomer here is no longer coupled to this moment here, then these now become linear equations. So this is just a constant. Now I have a series of linear equations. Linear equations, of course, have as solutions exponentials. So I can solve linear equations. So here's a solution for a linear equation. It's an exponential. And this, by the way, was actually first pointed out already in the 1980s by Bill Eaton and Frank Frone, who were working at NIH on a somewhat related problem, namely that of sickle hemoglobin aggregation. So this is the process that underlies sickle cell anemia. And so what they sort of pointed out that that really these equations were so intractable because of enforcing conservation of mass, that they really sort of pioneered looking at these linear solutions. So these are essentially the same kind of linear solutions as those that have been used by Bill Eaton and his colleagues to look at sickle hemoglobin. And of course, they match here. This is an exact numerically integrated solution to the master equation, and these solutions match here because we haven't depleted significant amounts of monomer. But of course, as soon as we get somewhere here, as soon as the reaction progresses to any significant amount, these solutions diverge.
How do you numerically integrate the master?
It's very straightforward. I mean, essentially,
Isn't it infinite?
right, but I mean, in principle, essentially what happens is that that's exactly right. But essentially what happens is that the equilibrium distribution is essentially exponential. So that's a very general feature of these types of linear systems. So therefore, you just define your accuracy, and that tells you how many up to what j you need to do your calculation for. So in principle, we solve. I think with this specific example, we probably looked at something like 50,000. Sort of independent polynomial numbers, but that's absolutely no problem to do using very standard algorithms. Okay, and in fact, one can check this as well using multi-part simulations, but it's essentially exact. All right, so now of course what one can do is one can take these linearized solutions, put them back in the equation, and solve again. So then you get the first order iterated self-consistent solution, which converges to the right sort of final value. So it's got rid of this sort of unphysical exponential divergence here. And typically, if you do this once or twice, you get something which is essentially within experimental error from the exact solution. So this is convenient in the sense that now we found a very easy to implement analytical scheme for taking these dynamic equations, master equations, and actually finding integrated dynamics, which is what we need in order to compare with experiments. And again, this is really nothing else than sort of these well-known Banach fixed point methods. And there's actually a variety of reasons why. Because you could ask, how do we know that this is actually a contraction? How do we know that we're actually going to converge? There's actually a variety of reasons for why that is, but it's related to the fact that the time evolution is actually well defined. So you can check that.
This M2 then is still in sort of explicit form as a function parameter.
Yeah. So I may have that on one of the. I certainly have an explicit expression of the M1 on one of my latest slides. So these are very easy. There's nothing. These are just combinations of elementary functions, basically. Nothing particularly exotic about these. Right. So another way of approaching these dynamics, which originated in sort of discussions with two fantastic graduate students a number of years ago, Sam Kern and Tuomas Knowles, is to sort of really look at these moment equations. So here, for the sake of argument, I've sort of simplified the system a little bit. So I've left out these sort of corrections that we discussed earlier. So really saying that the monomer is consumed by adding on to aggregates, and the number of aggregates changes either by new aggregates being formed, or in this case, secondary nucleation or fragmentation. So this is sort of a summary for secondary nucleation or fragmentation. So if I set this reaction order to 0, I have a process which does not depend on monomer, so that's fragmentation. And if I set this reaction order to be non-zero, I have something which is monomer dependent. Right, and so if we look at these equations, they sort of look somewhat similar to the Hamiltonian mechanics in the sense that I have two things, and I have sort of here one of them depends on some function of the other, and again here this depends on some function of the other. So this maybe reminds us a little bit of Hamiltonian mechanics. Let's try and do this in a little bit more quantitative way, and let's say, well, let's try and introduce some sort of canonical coordinates. So what happens if I say, let's think about momentum first. So what would be a good way to think about sort of momentum would be to say, well, what does momentum tell us? Momentum tells us what is sort of direction in which a system is evolving. That's momentum. So this system here is evolving depending on the number of ends. So let's define a momentum here according to this type of definition related to the number of ends. And it turns out that if you do that, you can show that there is a canonically conjugated position coordinate Q, which turns out to be the chemical potential. So it's a logarithm of the monomer concentration divided by the total available protein concentration. And if you do that and you define a Hamiltonian in the usual way, so a kinetic part p squared over 2 plus a potential where we need to choose this specific form for the potential, we actually get the time evolution of these moments from Hamilton's equation. So we differentiate h with respect to momentum, we get q dots, and p dot is minus dh over dq. Okay, why is that interesting? Well, because then you can say, are there any symmetries in this system? And of course there are symmetries, and the sort of one I will, the one that's sort of easy to see is, well, this is an autonomous system, so the Hamiltonian is time translationally invariant, and therefore there should be a quantity which corresponds to mechanical energy. Of course, it's not going to have units of energy or have an interpretation of energy, but formally there should be a quantity which is energy, which should be conserved, and indeed it should be the potential at t equals 0. And so is that actually true? So we're saying this sort of weird combination. So it's sort of slightly hard to believe, right? So this weird combination of sort of numbers of ends and masses of monomers which go into these exponentials, and with all these prefactors, we're saying that's conserved. So if we have, and that's very general. So we're saying any filamentous system, as long as it's an autonomous assembly system, then this slightly odd combination has to be conserved just because of symmetry. There's no other way. And so we amused ourselves by looking at some data. Some of this data is ours, some of this is from literature. In fact, these are all literature data, and in principle, we can extract these parameters from fitting various types of data. And these are very different systems. So these are, this is actin, so this is a functional filament system. This is the propagation of yeast prions, and this is amylin, so this is an amyloid system which forms in a variety of pathological conditions, including type 2 diabetes. So a very diverse set of molecular phenomena, very different proteins. The only thing that's in common is that they form filaments. And so in each of these cases, we take this sort of quantity which should be conserved, we plot it as a function of time, and indeed this is conserved.
So, I mean, I guess another piece that's maybe surprising from kind of a general nonlinear dynamics point of view is that, you know, even though this looks fairly complicated, it looks like you have just one fixed point, which is also why this fixed point theorem tends to work, right?
Yeah.
So is there a particular reason why you expect that you're just going to go to sort of one fraction?
Yeah. First of all, you can show formally that this operator is a contraction. So the answer is that because of the contraction principle, there's only going to be one fixed point. So formally, that's easy to show. And I guess what you're asking is physically why.
Yeah.
And I think one way to answer that question is that these systems are supposed to evolve towards a thermodynamic equilibrium, and that should not be dependent on the initial conditions. So therefore, at least part of these solutions should always be the same. So maybe that's one way of looking at it. But I think we don't. But I think we also don't know really why this scheme works so well. So typically, for many cases of practical interest, there's the first order self-consistent solution, just as sort of first order correction to linear solution, is sufficient to explain the data. So I of course chose a particularly bad example, so I could show the second order one. But typically, we just use the first order one. I think it's not entirely clear physically why it works so well, but it does. But I guess this is sort of a more. I think that's sort of interesting because it sort of gives you sort of a qualitative feel how sort of a correction to these linear equations. But I guess if you don't like that, then in principle you can all of this is exact, so you can just write down the dynamics and you.
But it looks then if you're basically saying your M1 on the previous slide, right? You're saying that also works really well.
Yeah.
So that seems to suggest that in some sense you have some sort of very rapid transition. that works well, if that's.
Yeah,
that's-
I mean,
That, and so that seems to suggest there's sort of some, sort of almost like a switch, some sort of hidden timescale in your problem where you have almost like a switch.
yeah. I mean, another way of looking at this is how many. In fact, I think that's that's another way of looking at it. You could ask how many physical timescales do you have in the problem, and it turns out that you're absolutely right. So in all of these, if you do sort of dimensionality analysis and you say I have whatever 8 3 parameters, how many reasonable independent timescales can I generate? It turns out that typically there's one which is actually important. And for example, as soon as you have fragmentation, this sort of timescale which defines the half-time, I'll actually come back to that because this underlies our ability to actually derive scaling laws for these kind of systems. And it turns out that for many cases, you only have one relevant timescale. That's another reason, probably physically, for why the self-consistent scheme works so well. And of course, these ideas which are sort of related to dynamic RG as well. As soon as you have sort of one dominant timescale, there's really a variety of ways in which one can approach this problem. But in any case, so this is sort of one can use these sort of tools from classical mechanics to really try and discover new sort of diagnostic tests for if you have if you have a particular system, you're interested in seeing whether or not it's really characterized by the formation of filaments. What is the test you can do? Is this quantity conserved?
Is the final state close to an equilibrium state, or is it a steady state?
Okay, that is a very good question. So I was not planning to expand on that in detail here, so I don't have any slides on that, so I'll just do a little bit of hands waving. So in principle, it depends. So there's a variety of ways in which one could look at that question. So typically, from an experimental point of view, so if one does an experiment, typically you don't reach a thermodynamic equilibrium on sort of any kind of reasonable experimental timescale, and that's partly due to the fact that you would need all the inverse processes.
Yes.
So you only get thermodynamic equilibrium when you satisfy detailed balance.
Right.
So essentially, you have to be able to sample.
You said you satisfied it for
Exactly.
association, dissociation. but not for-
Exactly. So for that one, so for the association and dissociation, for that one you actually do get
Close.
close
Yeah.
to an equilibrium. So for all the others you don't, and so you can actually include those in the master equation, no problem. You can solve the master equation even with all the inverse processes. Then when you look at the long time limit, you do actually find the equilibrium state. So no problem with that. Then when you compare with the experiment, you typically don't actually get that. And so that's the reason why I've actually left those processes out. So one could rephrase your question. So if I take the master equation I actually showed and I look at the T infinity limit, that doesn't satisfy detailed balance.
Yeah.
So that's not an equilibrium state.
I just think intuitively, they're all the time making filaments and they're getting longer and longer, and but it's the breaking of the filaments
exactly
that
right. So
limits that process.
That's exactly right. So essentially, you've now outlined a closed loop. So that you have
Yeah.
mass flowing from essentially things fragmenting, dissociating monomers, monomers adding onto fibers, fragmenting. So you have a closed
And then
loop.
those things will balance each other out. You'll get a steady state distribution.
Exactly. You get a steady state distribution, but you don't have detailed balance because you're only going around one way. And so that's why it is a steady state, but not thermodynamic equilibrium.
But I'm guessing it's very important you have the detailed balance on association and dissociation, so you won't get a.
Exactly. And I think that to some extent you also get a perfectly valid solution because the dynamics at early stages, the dissociation is not actually very relevant because you're so far from equilibrium. But that's the one process which is actually accessible in the lab in terms of the inverse process. That's where we're including it because our approach is to really, we're sort of quite close to experiments. In fact, the majority of my group and those of our collaborators are actually really sort of doing hands-on experiment. But in principle, there's nothing particularly. I don't think there's anything particularly profound about including those inverse processes. It gets a little bit more heavy in terms of the the size of the sort of expressions, but in principle, that does give you a perfectly reasonable thermodynamic equilibrium. We just leave it out just to make our life slightly easier because the dynamics doesn't play a role. Okay, perfect. So maybe sort of just one example of, in fact, we're running a little bit.
We started late, so.
We started late, so I might just skip to the sort of scaling laws because this is probably a slightly more general way of looking at this. So in principle, what one can show is one can take these integrated rate laws and sort of evaluate the, say, the half time. And it turns out that exactly in the same way as for the simple second order reaction that I started off with, the half time scale with the inverse monomer concentration. Now if you do the calculation, you do exactly the same calculation for these more complicated processes, and what you find is that the half time does, again, to a very good approximation, depend on the amount of initial reactants to some power. And it turns out that the power is related to the dominant mechanism by which you form new nuclei, and this is related to the sort of dominant time scale of the reaction. And so for primary nucleation, you get something which actually depends on the reaction order, so it's a reaction order divided by 2. For filaments which are mainly formed by fragmentation, it turns out you always get 0.5 independently of any of the other reaction orders or any of the rate constants. You always just get 0.5. And for the secondary nucleation, you get something which depends on the secondary nucleation rate, a reaction order plus 1 divided by 2. And it turns out that if you go into literature and you sort of ask the question of, well, what exactly does one observe? And so here again, we're looking at exactly this type of scaling relationship, so the relative lag phase or half time is a function of the chemical potential of the precursor monomer. It turns out that on this double logarithmic axis, of course, scaling laws will be linear relationships, and it turns out that the scaling law with an exponent of 0.5 is actually quite common. And this is interesting because this is a sublinear scaling law, so the exponent is less than 1. So if you thought about this in terms of classical nucleation theory, this wouldn't make any sense because this is not a meaningful nucleus size. So 0.5, half a molecule, that's not a meaningful nucleus. Whereas, of course, and in fact, there would also be no reason why all of these different systems from the literature for very different proteins under very different conditions would all have the same nucleus size. But here, by doing this kind of analysis, it emerges very naturally as a consequence of this type of mechanism. And maybe sort of one slightly more biological example, which is done together with our collaborators in Sweden, in particular Sarah Linz's group in Lund, University of Lund, is that of the AB, the peptide. So this is the peptide, the aggregation of which is associated with the onset and development of Alzheimer's disease. And so we thought it might be interesting to try and understand what the mechanism is by which it aggregates. And it turns out that this peptide does not follow this kind of universal scaling of 0.5. So it's not fragmentation based, so it does something else. And so, well, what could it do? Well, in principle, just based on the scaling laws, it would be compatible with primary nucleation or secondary nucleation. How do we know? Well, what we can do is, this is where having the rate laws is actually very convenient because we can start to fit to experimental data. And so these are curves here applied again, so we're looking at the fraction, the mass concentration of aggregates as a function of time, and at different concentrations. And the solid lines are essentially your best fit by assuming that all of the aggregates are formed by primary nucleation. And so here, this is really a global fit. So of course, we could fit each one of these curves individually, but that's not the point. We're describing the whole, have to describe dynamics of the whole system, not just in some part of the parameter space. And so this doesn't really seem to fit. And so the only physically meaningful mechanism that's left, so if we're saying the aggregates are not formed from just monomers, they're not formed from existing aggregates fragmenting, well, there's nothing else left except it has to involve both. And so, okay, so we say there's a great constant K2 for this, and there's some reaction over here. And then you get a reasonably good fit. And here, there's actually only 2 free parameters. So these are sort of related to the dominant time scale. So there's sort of one time scale for the formation of the aggregates initially, and this is a time scale for the multiplication of the aggregates. So this is actually quite a strong test. So we're fitting all of these data with only essentially 2 free parameters, and we get a reasonably good fit. So this is interesting in the sense that what this tells us is that these protein filaments A beta involved in Alzheimer's disease, they do grow according to these simple mechanisms. So monomers add to the ends, and first we form some aggregates, and they can grow by monomers adding to the ends. But as soon as we have any even small amount of these aggregates formed, then they will catalyze the formation of even more aggregates. So that's positive feedback. So the more of these I have, the easier it becomes to form even more. And so this is, of course, the way that you get runaway reactions, is if you have positive feedback. And so we think that this is not a coincidence that we're finding this kind of positive feedback in a process which is essentially associated with out-of-control protein aggregation. Yes, please.
The lengthening rate, the K plus,
yeah.
Do you re-seed with additional monomers to estimate that, or do you just do that directly from those curves that you showed on the previous
Great question, and you can do both. And so, in principle, the issue is that in the equations, the K plus is always connected to the number concentration of aggregates. So the easiest way to determine that parameter independently is to have a known, to actually have all reaction partners known, a known number concentration, known monomer concentration, and then you measure the rate. That's the easiest way to do it, so by using seeded aggregation.
Do you need some special structural characteristics of these proteins to get this branching structure?
The secondary nucleation. That's also a very good question which we've wondered about a lot, in the sense that it's not entirely clear why you would get a very specific secondary nucleation process here. So this is really new aggregates being formed just from existing aggregates, but it's a very specific process. If you take a protein where the primary sequence doesn't match that of the monomers, if I put something, where if this is a different protein from the green one, I don't have this process. So it's not just any surface, it's really very specifically the surface of these aggregates. And so there's different, so the structural basis is not known because we don't actually know the molecular level structures of most of these aggregates. But at some level, I think this almost reflects sort of a selection bias, in the sense that in order to have, so the systems we're very interested in, they're ones which are associated with disease states. And in order to have a sort of problematic aggregation reaction from a physical point of view, you probably need positive feedback, because otherwise it would be very easy for biological systems to deal with. If there's just a couple of aggregates forming, there's chaperone systems, etc. In biology, you can just remove these aggregates. In order to really have a runaway reaction, you probably need this positive feedback. And so we've looked at a range of pathological amyloid-forming structures, so not just this variant, but another variant which is also associated with Alzheimer's disease, the sequence 1 to 40, 1 to 42, in terms of the amino acids, but also alpha-synuclein, which is associated with Parkinson's disease. We also see this, and in fact, already in the 1980s, Bill Eaton, who I mentioned earlier, was looking at sickle hemoglobin aggregation, and he also found positive feedback for sickle hemoglobin aggregation. And again, this is a process once it's started inside the red blood cell, gets under control. So I think this is essentially a feature of pathological proteins. So proteins which cause trouble, they probably have to have this type of positive feedback. And there's a range of filaments which are functional. So if you look at functional filaments, actin, tubulin, etc., they definitely don't do that. So an actin filament doesn't catalyze the formation of more actin filaments from its surface, because of course that would be catastrophic for the cell. So there would be no way of controlling the sort of number of filaments. So I don't think there's any sort of, so it's certainly not the case that all protein filaments, nothing about the fact that you have a protein filament, that this will automatically give you positive feedback. I think it's really a specific feature of pathological proteins. But this is a conjecture so far. But of course you could say, well, if this is actually what's causing problems, well, why don't we try and turn this off and see what happens? And so how do you do that? Well, this is again where sort of theoretical approach can be very useful, because we can make predictions for what happens if we turn off particular types of processes. So this is essentially what happens in the absence of any inhibition. Here we're turning down the primary nucleation process, we get this kind of green profile. Here we're turning down elongation, we get a slightly different type of profile. And here we are inhibiting secondary nucleation. Right? So then what you can do, these are predictions, and now what you can do is we, together with our collaborators in Cambridge and Sweden, what we did is we looked at a wide variety of different species, and we found exactly one molecule turned out to be a chaperone, so Rico's chaperone associated with British dementia, and that matched perfectly with this essentially prediction of turning off the secondary nucleation. And you can actually do all of the sort of molecular level controls to show that that's actually what's happening. So you can show that the presence of the chaperone turns off the production of these low molecular weight species, oligomers, which are thought to be associated with toxicity. And indeed, in very biological experiments, if we now actually look at the effect, again, this is done with our collaborators in Sweden, if we look at the effect on brain slices, which can be kept essentially alive under in vitro conditions for long enough to do electrophysiology measurements. So collaborators looked at the power of these gamma oscillations, which is sort of measure the healthiness of the brain tissue. And if you now look at, so you expose this brain tissue to aggregates, which are formed in vitro, their healthiness decreases very dramatically, so these aggregates are toxic. And this is sort of ultimately what causes neuronal loss in Alzheimer's disease. But now if you take the exact same solution and you put these chaperones in solution, you still have the same amount of aggregates, or the same mass concentration of aggregates here formed at the end, but because we've circumvented this sort of positive feedback loop which produces all of the low molecular weight species, we've almost completely restored the toxicity. This is something which we're very keen to build on now in the context of the Center for Misfolding Diseases with my colleagues Michele Vendrusco and Christophe in chemistry. Yeah,
What's the assay on the right?
sorry, this one. Okay, so this is essentially looking at, essentially this is size, so this is monomeric species, the intensity of the, this is essentially a dot plot, so it's an immunochemical method for probing concentration of species, and they have been sorted out by size using size exclusion chromatography.
I'm only laughing because we tried to do that and we couldn't get that assay to work with a different amyloid system.
Right, right,
Yeah.
right. These are not easy experiments to do, and sort of a really big sort of, well, the credit goes to our collaborators in Sweden, Sarah Lindsay, who really sort of spent years optimizing these protocols. So these are definitely not easy experiments to do. So if I have another 5 minutes, I'd just like to show you a couple of examples of sort of physical measurements that we have been doing. I'll probably just show 2 examples really. And I'm going to focus on measurements in very small volumes. So we're going to be using microfluidics to do measurements in volumes which are around 1 million times smaller than those that are used in conventional biochemistry assays, where the sort of unit volume with which you work is microliters. Here we're working with essentially nanoliters, picoliters. Why is this useful? Well, one very simple idea is that if I'm interested in nucleation processes, if I study nucleation in bulk, this may be quite a difficult experiment to do because I have some number of nucleation events in my bulk system, and they will all happen at some different time. Probably very difficult to probe them directly because there's going to be very little signal independently of the method I'm using. There's going to be very little signal associated with a single event. And they're going to be mixed with signals from growth events. One way around this problem, which was actually already pointed out by Turnbull in the 1960s, it was a material scientist working at Harvard on a completely different problem, on the solidification of metals. And but he had conceptually a similar problem. So he was interested in the nucleation and the solidification of metals. And his solution was very simple. It was say, well, what happens if we just work in systems which are very small, and in particular smaller than the average separation between 2 independent nucleation events in bulk. So if we do that, we can be sure that anything that happens in any one of these systems is the result of one and only one nucleation event. In principle, we now manage to isolate these rare events, each one into their own compartment. So this worked fantastically in the 1980s for looking at, 1960s for looking at the solidification of metals. It still works today. The only problem is that the volumes that we have to use are really very small, so they are really of the order of picoliters. So the way we do this is by using droplet microfluidics. So we build devices like this in our lab. This is work that we're doing together with Dave White's group at Harvard. And so the way we do this is by using essentially standard soft lithography techniques. I'm happy to discuss if anyone's interested, but these are now becoming relatively routine techniques in order to pattern channels into various types of polymeric materials, which have dimensions of the order of a micron. So this is the channel dimension here, I think is about 15 microns. And then what we do is we flow an aqueous phase here, which contains proteins under supersaturated conditions, and we flow an immiscible oil phase, a fluorinated oil phase from the side. And of course, oil and water don't mix, and so what happens is you form at this junction here, you form thousands and thousands of droplets, as you can see here. So this video has been slowed down by a factor of 1,000. So we're using these lithographic techniques on the same chip. We can have these distribution channels and pattern these droplets onto sort of a larger area for observation. There's quite a lot of detailed chemistry that goes into sort of making sure that this sort of works. So for example, one has to use particular types of surfactants in order to stabilize these emulsions and make sure that the interfaces are but these are now becoming technologies which are sort of fairly routine to implement. So now what we can do is we can stop the flow, and we can measure what happens in this observation region. So I should say we've co-encapsulated a fluorescent marker molecule which lights up when it binds to the amyloid form of proteins, but not the monomeric form. So if we wait for long enough, this is now sped up by a factor of 100 roughly. We can see fluorescence appearing in each one of these droplets in the system. But if we zoom in on one of the droplets, the manner in which the fluorescence appears is actually quite interesting, in particular spatially non-uniform. So unfortunately we can only see half of the after the slide, but you can see sort of what happens here. Initially, we have no fluorescence, and then we have a single nucleation event here. And from the single nucleation event, we essentially have a spatial propagation throughout this droplet system. So somehow the formation of one of these aggregates here triggers the conversion of all of the other proteins in the system into aggregate form. And even though the size of this droplet is still much smaller than a bulk system would be, it's still much bigger than the size of any given aggregate. So this is not just one aggregate taking over the system, there's still thousands and thousands of aggregates, but they're all formed as a result of a single nucleation event. And this, of course, is the fragmentation of aggregates. Once you have one of them formed, you can break into two, they can diffuse and grow further and fragment, etc. And this, of course, gives reaction diffusion, and that's sort of what we're seeing here. So a reaction coupled to diffusion, and this gives rise to Fisher waves, which is what we're seeing in the system.
So you need positive feedback for this, and from the fragmentation or the secondary nucleation?
So both would work. So in this particular case, it's fragmentation. But in principle, in order to get a reaction diffusion mechanism in these types of systems, you do need a positive feedback.
You can predict the speed of these waves relatively easily.
You can. So the speed is related to the diffusion coefficients, and it's related to the reaction rates.
Turns out right.
It does turn out right, because of course you can measure the reaction rate independently. So from bulk kinetics, you get the reaction rate, and then then the unknown is the diffusion coefficient. And if you do the calculation of what kind of equivalent hydrodynamic radius by using Stokes-Einstein, what equivalent hydrodynamic radius you get sort of hundreds of nanometers the size of a system with these types of structures.
And what's the speed in micrometers per?
So essentially this is something which typically takes sort of an order of a couple of hours, something like that. And this is typically hundreds of microns or something like that. So in fact now, luckily it seems like the slide is showing, so this is actually these are actually a type of time scale, so hours, and we get hundreds of microns of propagation. This is in fact the equation, this is sort of the Fisher velocity, the wavefront, and it does only depend on the reaction rate and the diffusion coefficient.
What is the diffusion coefficient here? If it's 4 hours, it is very slow.
Well, I mean, so the rate depends on both, right? So you can have the diffusion, if the rate is slow, the diffusion coefficient can be fast, or if the rate is fast, the diffusion coefficient can be low. So it turns out that this rate here, this effective, says units of inverse time, and that's sort of related to the half-time of the reaction. And so that has sort of units of thousands of seconds. Then if you do the calculation, you get diffusion coefficients, which correspond to essentially supramolecular sizes, hundreds of nanometers. So that tells us that the species that's responsible for propagating this reaction is not a monomer. So otherwise it would propagate faster. So it's not a monomer, but rather an aggregated form of these molecules, which makes sense within this kind of mechanism where the propagation. Well, this type of, so this is the reaction diffusion equation. So this is the diffusive part, this is the reaction part, and the reaction part really comes from combining fragmentation with growth. So therefore, the diffusion coefficient should be the diffusion coefficient of one of these fragments. Okay, and so maybe, so I think the title of the seminar series was stochastic. There was the word stochastic, something to do with stochastic behavior in biology. So this is maybe just finish with this. So I haven't spoken about fluctuations. So the master equations that I showed were actually mean field master equations. There were no fluctuations. So what happens if we try and think a little bit about fluctuations? Well, we can actually write down a real master equation and say, well, what is the probability for observing? I have a given system. I want to observe n structures, total mass m at some time t. And again, I can write a master equation using exactly the same bookkeeping approaches, and I get something like this, where I'm considering only these processes. So I'm considering a primary nucleation process, where I go from a system which has n structures with mass m, and I'm creating one extra structure. So the number of structures is now n plus 1, and the mass is increased by the mass of one of these nuclei, say nc. And again, I can do the bookkeeping exercise, for example, for elongation. I don't change the number of aggregates, but I increase the mass of aggregates by 1, etc. So this results in this type of master equation. And so in principle we have these two variables here which characterize a system, the number and the mass. So that's a little bit inconvenient to deal with, but it turns out that there's actually a very good correlation between the number and the mass. So that's another way of stating that the average length becomes constant. And for fragmenting systems that's fairly easy to understand, because at some stage if a filament is growing at a steady state, then the probability of fragmenting will increase and then it will break and then this process will start again. So essentially I will get constant length. Turns out something similar happens with secondary nucleation. This allows us to simplify this and actually get exact solutions for these long times where we have this perfect correlation for the probability distribution. Again, this is work from fantastic, talented PhD student, Prof. Tuomas Knowles, which is just going to come out in PRL. So we really get these exact solutions for the probability distribution. And what's interesting, so it's interesting for a range of reasons. So it's interesting from a very practical point of view. So if I go back to the point I made earlier, if I'm interested in measuring a nucleation rate, so if I measure a bulk reaction here, I have lots of nuclei being formed at different times. If I ask the question, well, what happens if I change the nucleation rate by a factor of 10? I get sort of this green line here, it goes slightly faster, but because that's not the key timescale, the key timescale is a multiplication of the aggregates, it doesn't actually matter. If I make it 10 times slower, well, it's a little bit slower, and I have this green dashed line here. So so another way of looking at this is from bulk measurements, it's very difficult to determine nucleation rates. However, if I now look at the average time for an aggregate to appear in a system, it turns out that that's sort of roughly inverse in the system volume, and this is now very sensitive to the nucleation rate, because here I'm looking at the timescales for the formation of a single aggregate. And now if I do the same thing, I ask, well, what happens if the aggregates form 10 times faster? Well, I get something completely different. So from a very practical point of view, we like this, because this is one of the only ways I know of actually measuring in the lab reasonably accurately nucleation rates for these types of systems. And one can also actually ask the question of, in any one given system like this, how many nuclei are actually formed? And so you can make predictions because you have the exact solution, you have all the rate constants. It's slightly complicated by the fact that you may not be able to see all of the nuclei. So here, for example, is a system where I can see two independent nucleation events. This is time. I can see two independent nucleation events. But of course, if these events were too close to each other, the growth, this reaction diffusion mechanism, would obscure one of them. So you can ask the question, given that I know how rapidly these reaction fronts propagate, how many independent events would I expect to observe? And that's what we're showing here. And it turns out that this is actually more or less what you measure in actual experiments. So for the larger systems, you can see two or more events, and for smaller systems, you only start to see one. So we're now also increasingly getting a handle on the fluctuations. And of course, the further you go down to smaller system sizes, the bigger these fluctuations become. Okay, so I should probably stop somewhere around here. So just to sort of say that this was a sort of brief overview of a selection of the type of work we're involved in, sort of with a focus on trying to see how quantitative methods can help us actually understand molecular behavior, which is involved or underpins aberrant protein aggregation. And in particular, it allows us to sort of discover quite specific mechanisms. So for example, we discovered using this approach the aggregation of this A-beta peptide, which is associated with Alzheimer's disease, is actually driven by this secondary nucleation, this fiber-dependent nucleation mechanism. And I also showed you one example of how doing measurements in these small volumes, which is something we're increasingly keen on, allows us to really access key parameters like nucleation rates in ways which are actually just not possible using conventional bulk reactions. And I'd like to start by thanking all the people. So this is sort of collaborative work together with my colleagues in the Department of Chemistry here, Chris Dobson, filamentous, microfluidics work was largely done together with Dave Weiss's group at Harvard. The A-beta peptide work was all done together with Sarah Linz's group in Lund. And thank you very much for listening.